always wear gloves and a lab coat when working with pathogenic organisms.
Hi everyone, today we are going to discuss the first lab, and this lab is based on genetic engineering of E. coli cells with your gene of interest. So what we are going to do in this lab, that we are going to use E. coli cells, and remember, these cells must be competent cells. That means these cells will be made in such a way so that they can accept foreign DNA. Okay, so these cells are known as competent cells. So you take these E. coli cells and go through a process of heat shock transformation. This heat shock transformation is a very simple method of transformation where you take your cells, you take the DNA, and you incubate them at 42 degrees centigrade for about 45 seconds. Different labs do it for different times. We do 45 seconds in the lab. Then what happens in this shock, we call it heat shock, the plasmid DNA will enter the cells. Once they enter the cells, then you grow for about 45 minutes without any antibiotics. This is very important, that as soon as you do the heat shock, next 45 minutes, you should grow it without any antibiotics. At 37 degrees centigrade, this gives time for the bacteria, the E. coli, to replicate without any selection pressure. Okay? So after 45 minutes, what you need to do is you take the cells that you are growing and then you plate them on petri dish. Okay? Of course, in a laminar flow hood in sterile conditions. So when you plate them, be sure you are adding your favorite antibiotics to here. For example, in this case, we are adding kanamycin. Now be sure that the plasmid DNA that you are using, that also has the same antibiotics. Then you take this petri dish, store them in 37 degrees centigrade in an incubator. Do not shake, by the way. Store them in 37 degrees centigrade in an incubator, and overnight you incubate them and upside down. Okay. So next morning when you come, your plates look like this. So these are colonies of E. coli which are genetically engineered with your plasmid DNA. So what you do is you take a sterile pipette tip or sterile toothpick and then you pick up the colonies. And you grow the colonies in an LB broth media, again with antibiotics. So you grow it for 37 degrees centigrade overnight. And second day when you come back, you will have these cloudy cultures of E. coli cells. These E. coli cells, what you need to do is you do a plasmid extraction. Basically, you are extracting this plasmid which were trapped inside the cells. You do a plasmid extraction and then you run it on a gel after you do a restriction digestion. So you can choose your own restriction enzymes. For example, you can choose EcoR1 and Hindi3 to cut it, but be sure the plasmid maps have EcoR1 and Hindi3. You cannot just randomly choose any restriction enzymes. So be sure you follow the plasmid map. Once you cut the plasmid DNA with a restriction enzyme, you can run it on a gel. And if this banding pattern after cleaving or cutting matches with the plasmid DNA, then you know that your genetic transformation is successful. So this is a very simple method of genetic engineering of E. coli with a plasmid DNA. Thank you.
Take your cryoculture out of the -80 freezer and make sure to keep it on ice or in a cold block. In this lab we will be using DH5-alpha strain of E. coli, which is not yet competent. It is important to thaw the culture slowly, so do so on ice. It will take about 30 minutes. Next steps will be performed in the laminar flow hood. Disinfect it with 70% ethanol before you start. Have your sterile media ready. We will be using 2 tubes of LB, 5 ml each. Anything that goes inside the laminar flow hood must be disinfected, including your hands, containers, and tools. Once your work environment is sterile, attach a tip to your pipette. Carefully open your cryoculture tube. And briefly touch the cryoculture with a tip. Then open your media as shown, and again touch the surface of the liquid with your pipette tip. This tube is now in inoculated culture. Liquid media cultures are to be grown in a shaker incubator. Place the tube at an angle to allow sufficient aeration. Set the incubator to the right temperature and shake in RPM. And make sure the machine is running. Take note of the time and date to properly document your experiment. Next day, the cultures should be visibly turbid. This means you're ready to start the next step. For this part, you're gonna need ice. Stick your cultures into it to keep them cold. Prepare your pipette and the corresponding tips, and 2 sterile microfuge tubes. Disinfect the area by generously spraying with 70% ethanol. Next, you'll need to transfer about 1 to 2 milliliters of your culture into each of your microfuge tubes. If you have 2 separate cultures, for example labeled A and B, then label your microfuge tubes also A and B and transfer correspondingly. Make sure to pipe it slowly. You don't want to damage your E. coli by forcing them through the opening of the tip at high pressure. There, I finished transferring culture A into microfuge tube A. Now we'll do the same for tube B. Also keep both your main cultures and your microfuge aliquots on ice. For the centrifuging step, make sure your cooled centrifuge has reached the right temperature, usually +4 Celsius. Also, of course, set it to the right RPM and time. Touch the rotor with your finger just to make sure it's actually cold. Don't forget the inner lid. And begin. When the centrifuge is done, retrieve your samples. Spray them with ethanol to disinfect, and put them on ice right away. Next, you will need to discard the supernatant while leaving the pellet on the bottom of the tube. You can do it either by aspirating with a pipette or just decanting like I do in the video. Now, again, fill your microfuge tubes with corresponding cultures. Together with centrifugation and the step where you discard the supernatant, this process will be repeated until you're out of culture. The purpose of this cycle is to collect all the bacteria you have in the culture as a pellet on the bottom of the tube. When your cultures run out, you will need to fill the tubes with calcium chloride for the final centrifugation step. As our competent cells will be stored at -80, we need to resuspend them on 15% glycerol in addition to calcium chloride. If it's frozen, let it thaw at room temperature. Also prepare a cryoculture tube. They look like this with a different kind of bottom. Get your samples from the centrifuge. Make sure your glycerol and calcium chloride solution is in a liquid state. Now discard the calcium chloride supernatant just like you did before. And same technique, add about 1 milliliter of 15% glycerol and calcium chloride solution to each one of your tubes containing the pellets. Try to gently resuspend by flicking the tube. This usually doesn't work though after multiple rounds of centrifugation. The other option is to resuspend by pipetting up and down. Be gentle and move the plunger very slowly. Aspirate and dispense the liquid near the pellet several times to try to break it up. You can try to dispense while the tip is puncturing the pellet. That should break it up from within. Also, slightly swirling the tip inside the tube might help. You are done when there is no pellet left and instead the entire liquid turns turbid. Flicking the tube again might ensure a more even distribution. The pellet still doesn't seem completely broken, but it should not result in any significant problems. Now, transfer the entire content of the microfuge tube into the corresponding cryo tube. And do the entire process again with the other sample, starting with resuspension. And now your competent cells are ready to be placed in the -80 freezer for storage. Make sure to properly label your cryocultures. Get your samples from the -80 like before and make sure to keep them on ice. Once again, ensure sterile conditions. Prepare your solid media with the right antibiotic and set it aside for now. Get your plasmid DNA. It must be pure and concentrated. Have all your necessary tools handy and sterile. And you will need a fresh microfuge tube for each one of your cultures. Pipette tube boxes should be conveniently arranged and, you guessed it, sterile. Adjust your pipette to the volume that you're going to transfer. For us, it's going to be 20 to 50 microliters. And perform the actual transfer. Cryoculture A to tube A, cryoculture B to tube B. Then, add your plasmid. You can calculate the right volume from the concentration of your DNA. Flick your DNA tube to make sure its concentration is even throughout the solution. And do the same for the other sample. Gently flick your cultures with DNA before letting them incubate on ice. Meanwhile, turn on the instrument that you're going to use for heat shock and set it to the right temperature. When ready, make sure the temperature is correct and begin heat shock. Make sure to time it precisely to about a second. When time is up, immediately transfer samples to ice. For the next step, we will use SOC. Watch out, it's very easy to contaminate. All you need to do with SOC is add a certain amount of it to each of the cultures. These cultures will need to be outgrown in a shaking incubator. If the incubator you're using doesn't have racks for microfuge tubes, then just tape them up somewhere inside at an angle. Set the same temperature and RPM as before. Take your cultures back to the laminar flow hood and put them on the ice. The last step is to inoculate solid media in a petri dish with your cultures. I split my plate into 2 zones so that I can use one plate for both of my cultures. For this part, you simply aspirate all of your SOC culture and dispense it on the right part of the petri dish. Now you need to spread your E. coli. You would usually do it once over the entire dish, but I'm doing it individually for each side A and B. If you're not familiar with this technique, you will soon get to know it from other classes. The last step is to wrap the edges of your dish with paraffin. Start in one place and go all the way around, trying to make it as airtight as possible. This plate should be incubated in a normal, non-shaking incubator at the same temperature as you used previously. In the next few days, you should begin to see colonies on your plate. Pick a few that are separate from the others and are of simple round shape. Misshaped spots might actually be clusters of small colonies. You don't want to have that in the culture. Use your selected colonies to inoculate cultures of liquid media. It is the same procedure you've seen earlier in the video, except this time you will need to add your antibiotic of choice. For our strain of E. coli and vector type, we use kanamycin. It is usually filter sterilized and added to your media right before inoculation. When you have your dense overnight culture, you will need to extract the actual plasmid DNA from it before you can proceed to the next steps. This is usually done using various proprietary extraction kits. The exact procedure will depend on which particular brand and type of kit you're using. Your output will be a large amount of DNA suspended in a small volume of buffer. DNA concentration and purity of this solution is usually evaluated using a special spectrophotometer called nanodrop. It is easy to use and you can find demonstrations online. Uncontaminated DNA will produce this kind of curve. And you get a simple readout of your DNA concentration. This number will help us calculate compositions for the restriction digestion reactions. These are also done using proprietary enzymes, so you will have to look up the right protocol. Let's say we choose enzymes EcoR1 and Hindi3. They will cut your vector at those three positions. Now, to estimate the fragment sizes from a map like this, imagine a clock face overlaid on top of it. The shortest fragment on the top appears to span about 1 hour. Divide that by the total of 12 hours and multiply it by the size of your vector. And we get about 1 kilobase pair for this fragment. You need to know sizes of all of your fragments before proceeding. Note that a lot of the time you can find the complete map of your vector with the position numbers indicated. Only use the shown estimation technique when you cannot do so. In the last step, we estimate the sizes of the fragments of your vector using gel electrophoresis. Don't forget to zero your balance with the weighing dish on it. To prepare your gel, use only molecular biology grade agarose. For most applications, your gel needs to be about 1% concentrated. That is 1 gram of agarose per 100 milliliters of TAE buffer. Our gel will be about 70 milliliters in volume. TAE buffer tends to precipitate over time, so make sure you mix it up. I usually just invert the vessel. Prepare your measuring glassware. Measure out the right volume of a TAE buffer and have it ready. Get a vessel that you're going to mix your gel in and add your agarose first. You can pour some buffer into your weighing dish to help get all of the agarose powder out. And put the rest of the TAE into your mixing vessel. Crumple up some tissue and use it to plug the neck of your vessel. Then put it in the microwave and start heating. You want to stop right at the point when your solution starts bubbling. Get your vessel out. Be careful, it's hot. Use heat-resistant gloves or some kind of tool to pick up the vessel. And gently swirl it until it's not too hot to touch. The solution is cool enough when you can hold your hand against the bottom of the vessel for about 5 seconds without hurting yourself. Get a pipette and the matching tips, and prepare your dye. This dye is extremely carcinogenic, so wear gloves and don't let it get on your skin. It's also photosensitive, so try to minimize the time it spends outside of the box. Set your pipette to the correct volume. This particular dye is 10,000x, so we only need 7 microliters for 70 milliliters of the TAE buffer. Add it to your solution. Put the dye back in the box. And dispose of the tip in the green bin since it's now contaminated with a carcinogen. Prepare a cast for your gel. This is actually what determined the volume of TAE buffer that we needed previously. Equip it with a comb, it will shape your loading wells. Then get your agar rose, swirl it a bit, and gently pour it in the cast, making sure to avoid any bubbles. And let the gel rest until it fully solidifies. Meanwhile, prepare your samples for loading. You will need first your sample, loading die, this one is 6x, and some kind of vessel to mix the two together. Let's say I have 25 microliters of my sample left. I'm going to mix it up. And add it to my mixing vessel. Now, this particular dye is 6x, so for 25 microliters of my sample, I would need 5 microliters of the dye. This varies depending on the manufacturer and type of the dye. Draw the dye slowly, it has some viscosity to it. Add it to your mixing vessel and pipe it up and down a few times to mix it well with the DNA. Also mix by flicking. It is important that all the DNA molecules in your sample make contact with the dye. By now your gel should be ready. Touch it to make sure it's solid and carefully remove the comb. Unlock your cast. Remove the tray. Gel should be stuck to it. Place it into the electrophoresis chamber with the wells being on the same side as your anode, here indicated as the black terminal, and away from the red terminal. Fill the chamber with TAE buffer until it fully covers the gel. The gel should be right below the surface of the buffer. And finally, we need to load the gel. I usually start with a ladder. You need to carefully reach right into the well with your pipette tip. Dispense the liquid slowly and it should go right to the bottom of the well. And do this for each one of your samples. Take care not to pierce the sides or the bottom of the well with the pipette tip. This is what a loaded well looks like from a different angle. Connect your electrophoresis vessel to the power supply. With most equipment, the right terminals fall in place automatically. Turn on and set up the power supply. The standard is 90 volts for 90 minutes. And make sure the power supply achieves constant voltage. Easy way to check if your gel is set up in the right direction is to remember that DNA runs away from the bubbles and it should run into the gel. When done, our gel should look like this. Note that the wells are on the left side. Now you can use the DNA ladder to estimate the size of your bands and confirm or deny the presence of your plasmid.